Assalamualaikum and hi to Madam Sarina. My name is Nur Khalida binti Shahlan. So for this time, my group mates and I will be talking about the research on the Sharia Tambagaya tree, an endangered species found in southern India. As stated in the article, this tree is valuable for its fine woods and medicinal qualities, but habitat loss and misuse of it are putting it in danger. So in order to protect and conserve this tree, this study makes use of the plant tissue culture method. Due to significant seed predation and limited seed germination, traditional tree growing techniques are ineffective. So we can safely cultivate large numbers of healthy, disease-free plants thanks to tissue culture.
The research starts with plant collection and preparation. Healthy mature fruits were collected during April and May around India. The endocap of the fruit were removed to get the seed. Then, the seed were washed in 5% 2020 and 70% Sevlon before treatment by antifungal bavistine and rinsed 4 to 5 times with autoclave distilled water. Next, Terra with 0.1 HgCl2 and rinsed again before blow dried. Second step, in vitro germination. Seed will germinate on half-strength MS medium without plant regulator. The pH of 5.8 and additional ascorbic acid and activate charcoal. After 4 weeks, the germination was recorded. Next, shoot induction and proliferation. Shoot tips were initially cultured on half-strength MS medium, sweet sucrose, and agar for 2 weeks. The medium was then shifted to full-strength MS medium with sucrose and agar supplement with different concentration of cytokinin regulator. Morphogenic response, number of expand, and the number of shoot buds induced were measured. Routine process. Multiple shoot were cultured on half-strength MS medium alone or supplement with auxin regulator. The culture will maintain at 24 to 28 degrees Celsius under 16 light photo period. Acclimatization and establishment of plantlets in soils. Rooted plantlets were rinsed with sterile water and submerged in bavistine before transplant in pot containing the mixture of garden soil, compost, and vermiculi. The potted plantlet were maintained in a greenhouse, then were transferred to 18 pots with garden soils for further development. The DNA isolation and genetic fidelity analysis genomic DNA from one mother plant and 9 in vitro grown plants were isolated using the CTAV method and checked with agarose gel electrophoresis and nanodrop. PCR amplification was done and the product were visualized under UV light. The bands were scored to calculate the Jaggard similarity coefficient. Finally, experimental design and statically analysis. In vitro proliferation, experiment used a completely randomized design with an at least 50 seeds and 20 shoots per treatment. Data were collected every 7 days and analyzed using two-way ANOVA and TKHSD test with SPSS. The genetic association between mother and in vitro plants were assessed using the Jaggard similarity coefficient with NTSYS-PC.
Indole 3-butyric acid (IBA), a hormone that promotes root development, has been found to be effective for Shorea tambagaya, with similar results observed in related species. The best method for inducing root growth involved using well-established shoots in a nutrient solution containing varying concentration of root-promoting hormones. The ideal concentration of IBA for optimal root growth is 4.48 micromoles per liter. Next, to determine if the DNA of the plants developed in the lab micropropagated plants was identical to that of the original plants, which is mother plants, scientists employ a technique known as ISSR markers. So the researchers analyzed the DNA of lab-grown plants by using ISSR markers and discovered that there were only slight variation from the original plants. This suggests that the lab-grown plants are genetically stable and comparable to the original plants, though the tissue culture process may still cause small genetic alteration. Moreover, further confirming the results of earlier research on several species, the study showed that lower concentration of naphthalene acetic acid and AA were more successful for root induction.
Next finding is regarding morphological similarity. The rooted plantlets generated from seedlings appeared morphologically similar to the plantlets from the ex-plant collection site, indicating the success of the in vitro propagation protocol. Therefore, the morphological characters of Viola pubifolia fall well within the range of variability of all these species. And then we have primer sequences and genetic analysis. The profiling showed little genetic disparity after in vitro proliferation, in which these primers were used, scoring 24 monomorphic and 6 polymorphic bands, while ISSR markers were used to assess genetic diversity in the stock C, generating 48 bands and a polymorphism rate of 87.5%. And lastly, about the finding is conservation efforts. So this effort actually helped to develop an efficient in vitro propagation protocol for Shorea tambagaya. Low genetic diversity and heterozygosity in V. epipsilla populations may explain its low tolerance to change the environments and risk of extinction. Declining populations and habitat loss suggest conservation strategies are needed, in which increased interspecific hybrids between Viola epipsilla and Viola palustris indicate anthropogenic changes in peatlands. In conclusion, the research study successfully developed a tissue culture-based microcloning method for this species, in which this method can be used to mass produce high-quality plantlets for conservation and restoration efforts.